kind of dying, so we'll make that our circular labeler thingy. Anything with the word cyto in it means what? Cell. Good. Go ahead and just know that we know that. What are the three cytoskeletal elements? And if you don't mind, I know that this is a little bit different. Because this is being recorded, just whenever you write something, just take 2 steps back or whatever, and then come back in. I know it's kind of irritating, but it's okay. So yeah, what are the 3 cytoskeletal elements? Okay. They're really poorly named, but can you name one of them?
Like elements, like what's in it?
Yeah, or just what we call them. It's 3 proteins. Yeah, so yeah, there's 3 of them. There's the microtubules, there's the microfilaments,
and the intermediate filaments.
Intermediate filaments, right? Really irritating diction though, right?
Yeah.
But yeah, those are the 3 that we want. Microfilaments, right? Yeah. So for these 3 cytoskeletal proteins, we're going to classify them based off of their functional role, and that's kind of a long list of things we'll talk about, but then their actual substructures, what they're made out of. So let's start with microtubules because that's really the most extensive one. So we're just going to really just define their functional role here, because that's going to be a whole set of its own map by itself. So function.
Like what function? They transport stuff. They move it.
Yeah, they just provide a highway for transport vesicles. But we're for this map, because there's a lot of things that we're going to say. We're going to talk about their role as structural proteins, transportation highway, and then you probably also learned that they play a role in cilia and flagellum and other things. So we're going to have an individual map here for function of, in this case, microtubules. So we're just going to say function there. Okay. So yeah. I know. So you listed some of the functions. What are some functions that you can list of microtubules?
They transport cells. I don't know if I know cilia and flagellum.
Okay.
They just transport.
If I were to say that you have a cell that's really round and globular means round. It's a weird word for ball. It's really round. If I were to suck out all the microtubules inside a cell, would it still be round? It would?
I don't know, because it's structure. It helps with structure.
Well, let me give you an anthropic example. If I were to suck the bones out of you, you wouldn't still be standing up, right? You'd be this blob thing so microtubules specifically play a really large role in providing the shape, the morphology of the cell. If a cell, have you ever seen like a brain cell by any chance?
Mm-hmm.
Okay. Brain cells have this really like spiky star-looking pattern to them, and the reason that they have the shape that they have is because of the arrangement of their microtubules. Have you seen what like maybe certain epithelial cells look like? They're really flat and thin, and they have the shapes that they have because of their microtubule arrangement. The others too play a role as well in terms of like telling you what the shape of the cell is going to be, but it's mostly the microtubules. So one of the functions of microtubules is, we would say, the morphology, and that's the word that we would use for that. Good, good. Just to give you an example, anything with the word morph, morphogen, morphos means shape. Again, a lot of biology is just a redundant form of nomenclature. Okay, good job. So you had mentioned what else? So we have that they give you the shape, and we'll talk about why that is later. But what else do they do functionally?
Transport.
Transport. So they provide a framework for a intracellular transport. So, we'll just do transport. We'll talk about the two types later. Good job. So what are the, when we say transport, that's kind of an ambiguous term. If I wanted to send, let's just say this room is a cell, okay? And I want, and I'm the mitochondria, and I need to send something, or let's be more specific with what you guys have been going over. Let's say that I am a Golgi body, and I want to excrete something on the other side of the cell. How am I going to send a signal from here to the other end of the cell?
The microtubule kind of thing.
Good, good. So we say transport, vesicular transport is what we talked about. So for transport, we're going to have another arrow for vesicular. And you could also sometimes say intracellular if you wanted to. Transport for things within the cell. Good job. And then what's another type of transport that we have?
Is it a secretion type of transport? No, right?
Well, yeah, that's kind of a ambiguous terminology there. Let's say a sperm cell. How does a sperm cell get from one point to another? If you don't know, I mean, yeah, okay. If you see videos of them, you'll see them that they actually kind of have this like dolphin tail thing at the end of it, and that helps them move from one point to the next. So maybe you've seen like those famous pictures that they have of a sperm cell fertilizing an egg. Those are what would be called extracellular transport. And what I mean by that is that the cell itself is moving. We're not moving stuff inside the cell itself is moving. So extracellular. Okay, so for extracellular transport, we just mentioned one of those, which was the what? Sperm cell, and that's called a, whenever microtubules are contracting to move something, we call that a what? So again, the microtubules are moving the cells themselves, we call that something called a flagella. That's what we say. And so, let me ask you this, and I guarantee you he will ask you one of these questions, because he's a big guy, he stresses structure and function a lot. Is it a propeller, or is it a dolphin tail, kind of a whip thing? Which do you think?
Flagella?
Yeah. For eukaryotic flagella. Good, dolphin tail. Bacteria use a propeller, good. The reason why he mentions that, and there are examples in developmental biology, you'll learn about certain types of cilia that do this type of thing, but a circular flagellum is doing a propeller-like motion is structurally very different than what you see here with this type of tail-whip motion. Just letting you know that. So we have flagella. What are some other examples that he may have mentioned in class that you remember?
Well, we just talked about cilia and flagella together, so the wouldn't be cilia.
Cilia, right? What would be, how does cilia work exactly? How do they move? Well, no, they still have the same bending back and forth type of tail whip motion, but does a cilia move the individual cell? No. Did he give an example of where the cilia are found in the body?
No, we just talked about flagella and sperm cells.
So flagella and sperm cells, let me give you an example, and this is a great example here. There you go. Coughing, right? Cilia, and let's just be realistic. By the time air reaches your lungs, it's relatively sterile. Relatively sterile, key point there. As it goes through here though, just taste the air here, right? There's dirt in the air, really bad, right? But nevertheless, by the time it reaches your lungs, it's sterile. So as air flows through your lungs, it's gonna come into contact with mucosa, mucosal epithelial that have cilia in it. And the cilia are gonna do a really good job of trapping that dirt, those irritants in that air, and then moving it up through your throat where you're going to either cough it out or spit it. You do this thousands of times a day, you're not even aware of it. Have you ever been to a concert?
Mm-hmm.
Was there crowd surfing? Do kids still crowd surf?
Sometimes.
Okay, so in crowd surfing, if someone's crowd surfing, I move them away from me. I don't move. I move that person that's away from crowd surfing. That's kind of the same thing that's happening with the cilia. The cilia are basically crowd surfing your phlegm, in this case, out of your throat, where you can either expel it. Another example would be the fallopian tubes in female reproduction. They have a lot of cilia that creates a current that sucks in that secondary oocyte. So those are just two examples, but I want you to understand that the cell itself doesn't move. It's moving stuff around it. So yeah.
So flagella move, like the cell itself moves and so the cell doesn't.
Mhm. Good job. Okay. Let's just talk a little bit about, I'm just going to talk about it, I'm not going to make you write it, but the shape. What shape do microtubules have? Good, they're cylindrical. Because they're cylindrical, they resist something called compression. And there's really no easy way to explain this, but it'll make sense when we talk about the intermediate filaments and the microfilaments. So let's just go ahead and just add one more function to this. And we're going to say right here, resists compression. As a function of it, yeah. Good job. And just so that you know, it's because the cylindrical shape maximizes the cross-sectional surface area, and the cross-sectional surface area of something is what determines how strong it is. And so that's how we do that. Let's go ahead and just circle these though, and then fill in our maps. I think that marker's dead. We'll use red, I guess, for connecting the points. So you're going to have 3 arrows branching off the function, one there, and then yeah, one there. And you can go ahead and just circle these terms here. Go ahead and circle cilia and flagella. I know I'm being picky here, but we'll go more into that there. Okay, so that's their functional role. Let's talk about their structure. What are they structurally made out of? Okay, that's fine. So we'll go ahead and around here, we're going to define the structure. Okay, so there's lots of ways that we could say this and lots of things that we could use to define this, but I'm going to define them in terms of their large structures and then their small structures. And I'm actually going to go ahead and provide us with 2 arrows for that. So we're going to have one here saying small, and that won't take up too much space. And then the other one will be large. Yeah. So at the small scale, what I mean by that is, if I took a microtubule and broke it down into its individual parts, what would that individual part be?
Like what it's made of, or like.
Yeah, what it's made of. What are they made out of?
I mean, they contain, well,
Kinesins interact with them, yes,
Yeah.
as do dyneins, and we'll
Yeah.
explain what that is here in a sec. But for the structure of it, they're made up of a protein. Do you know what that protein is called? Okay, it's called tubulin.
Mm-hmm.
That's the protein. So small level structures is just tubulin subunits. Why does it end in IN?
Because it's a type of protein.
Good. Anything that ends in IN, tubulin, insulin, myosin,
It's a protein.
is a protein. Good. I like that. At the large scale structures, what are the structures that we can have in terms of the larger scales of microtubules? Okay, so are cilia and flagella just individual microtubules, and does it form one giant cylinder, or is there a specific arrangement that they have?
They have specific arrangements.
Good. What are those specific arrangements?
So they have cells, or they don't have cells.
Yes. But what are the, I think, do you have your notes with you? Look in your notes and make sure that he mentions this.
2 plus 2.
Yeah, the 9x2 pattern is kind of important. That forms kind of the larger structures like cilia and flagella. Some people use a 9+0 pattern for the basal body, but I don't think that's super important unless you mentioned it, did he?
He mentioned the 9, there was one with 9 times 3, or, yeah, 9 times 3.
Yeah, I wouldn't, well, we can go over that and we can touch up on it, but yeah, that would be the basal body in this context would be that. That's slightly important, but I really want you to understand that anytime you have something that is, let's say a flagellum, it has a 9+2 pattern, or in a cilia it has a 9+2 pattern as well.
So the large ones have a 9 times 2 plus 2.
Yeah, so for the large, what we're talking about here is really just the two substructures that we have, but we'll say the 9+2 as one, and then the 9+3 is the other. Yeah, I think that's a 9 by 3 subunit 0. It doesn't really matter. As long as you have a picture of it and you're available to understand it, that's what really is irritating me. One of these, let me just pull up actually. I don't think I have it with me. Okay, back over here. So the 9+2 pattern is what? It forms the flagella and the cilia. So we'll just draw an arrow kind of connecting this to here so that we know, okay, that pattern there is present in that. You could, yeah. I mean, it's kind of a little bit ugly, but it's all right. Who cares? As long as it's functional and you understand the relationships that are representing, structure determines the function. That's what I want you to know. For the 9x3 pattern, this makes something called, they call it the basal body, but also the centrioles. So let's put that on there. Why are centrioles important?
Aren't they like chromosomes?
No, but they play a role in chromosomes, but they're not stored within. Okay, so centrioles, if I have a bunch of centrioles together, what do they form? Okay, what we'll do is we'll just draw another arrow down here. If I have a bunch of things coming together, there's an ending in biology that we use for that. Do you know what a ribosome is?
Mm-hmm. And it helps make proteins and stuff like that.
Good, yeah, it helps make protein. Do you know what all it's made out of, though?
Mm.
It's made out of a lot of things, right? They're not super large, but they're not just one specific biomolecule. They're a mixture of RNA and protein. Do you know what a chromosome is?
Yes, we have 46 here.
Yeah, we do, right? But do you know what they're made out of chemically? Yeah, that's okay. They're a mixture of DNA and protein. Desmosomes, ribosomes, chromosomes, polysomes, microsomes.
They're a mix of stuff in DNA.
Good. Anything that ends with the word some means that it's a bunch of things in biology that are all coming together to form one really big process. Some people, when they talk about cell replication, call it the replisome. You can add some to anything, and that's what that means. So centrioles come together to form something called a centrosome. Okay, good. And the function of the centrosome is to do something called the microtubule organizing center. Have you heard of that?
Mm-hmm.
Okay, so what we'll do is we'll actually include this over here as well. As a part of the centrioles, because the centrioles are the functional unit, but if you have a bunch of centrioles together forming a centrosome, you have a microtubule organizing center. Organizing center. Sometimes called the MOC for short. Yeah, microtubule NTOC, yeah. What's in a name? Okay, so maybe let's just draw an arrow kind of connecting these two to say that they're synonymous terms. One of those terms says what it does, which is organizing microtubules. Another one describes what it's made out of, which is centrosome, which means sewn a lot of centrioles together. Really irritating, redundant kind of nomenclature, right? Like, why do they have to have the terms that they have that they do? Okay, that is pretty much it for microtubules that I have at this point. Let's talk a little bit down here about vesicular or intracellular transport. And then we'll also talk a little bit more about extracellular or flagella type transport. So for transport, let's just draw one arrow here that kind of illustrates what we're talking about. And this will make sense later. So, how do I move something in the case of either vesicular transport or extracellular transport? There's a class of proteins that we use for this. What are those type of proteins called?
That are on the microtubule?
That interact with microtubules and help things move.
Like kinesins.
Well, kinesins would be one of them, but what is the other one?
Dyneins.
Dyneins. But what are they all in a big super family of what? Right? It's so irritating. Motor proteins is what we call them. So motor proteins are the name of the game when we're talking about transport. And you had already mentioned, what are the two types of motor proteins? Dyneins and kinesins. Yeah. Wonderful. Good job. That's okay. I am so bad. Let me give you an example. I spelled parallel with two R's. I spelled balance with two L's for the longest time because the cerebellum has two L's in it, and the cerebellum gives us balance. So I spelled it with two L's. I'm really bad at spelling, by the way. Let's just talk about the dynein. That's not really helpful nomenclature, but kinesin. What does the kine mean?
It's like kinesiology. It's like
Yeah, kinesiology is what?
violence, movement, and
Yes, movement, good, right? Anything with kine in it means movement. Kinetic energy is what?
energy, movement.
Energy, emotion, right? So anything with a kinesin, we're talking about a protein, because it ends in IN, that is involved in movement. What do kinesins play a role in moving?
And well, they're on the microtubule. They're kind of like little legs.
Yeah. Did he show you guys the video from like Harvard did a long time? You can actually see the kinesins,
Well,
sorry.
He didn't show us. The SI leader showed us.
Oh, okay. Well, there's some really cool videos out there that people like at MIT or Harvard have done, where they you can see a video of a kinesin motor protein walking, and it literally looks and it moves as if it was walking, and that's really creepy.
And
that it.
The kinesins look like they're walking, and then this one kind of like does like a shuffling thing.
Yeah. It looks like it causes the spinning motion back and forth. So wonderful that we know this. They both are hydrolyzing ATP. We'll talk about what ATP means later, though, because I don't think has he explained to you guys what ATP is and why it's important?
We know like that they use ATP instead of energy, but like I don't remember going like what is ATP specifically.
Okay. He'll probably stress on that stuff later, but you obviously know that anytime we need to do something in a cell, we use ATP for this. Let's just classify these because they're motors, you can imagine, on what they move. So what do kinesins move?
I know they move, like they help with moving mRNA, and they can help move like the from head to something like that.
Well, so we haven't talked about it, but I know that in your class he talked about the process of making a protein. That we have
proteins.
We have vesicles that transport stuff from, let's say, the ER to the Golgi bodies.
Yeah.
So kinesins are one of those things that move vesicular transport.
So like,
Yeah. Just draw an arrow connecting vesicular. Kinesins are the ones that are involved in vesicular transport. One more thing that we want to mention about kinesins is that they can play a role in the movement of organelles. So let's just say that you're, I don't know, let's say a T cell, and you're about to unload a very dangerous payload on a cancer cell. What you will do is you will use the kinesins and other microtubule rearrangements to actually move the Golgi complex so that whenever those proteins are going to be released, they just go in a straight line to wherever that stimulus is. So that's kind of creepy to me that cells can orient themselves in 3 dimensions. That's really weird. Anyways, what do the dyneans do?
Well, they also have to transport. We really live on that kind of.
That's okay. So kinesins literally will walk along microtubules.
Yeah.
What dyneans will do is they will grab a hold of 2 microtubules, and one of them will contract and then relax, and then the other one will contract and then relax. And through this, they can create this kind of bending motion. And that bending motion is what the cilia and the flagella use to move. So let's just draw 2 arrows connecting the cilia and the flagella to the dyneans. Good job. Okay. Have you used the app for the smartboard?
Mm-hmm.
Is it helpful? Is it saving the information in ways that you can
It's
view
like
it?
pictures. So like
It's
I would have to
pictures.
take a photo of it, and like I just push like the button. It doesn't record.
Oh, it doesn't record, so you can't like say what I did this. Okay, if you want to take a picture of this, you can, because we're about to play review. And if you don't mind letting me know how we're doing on time, that'd be helpful as well.
We have 32 minutes.
1. Okay, did you get a picture of it?
Mhm.
Okay, now I'm gonna erase things. Okay. Come on up here and explain to me what these are. So start with the function. Tell me the function of microtubules.
Well, the function is to transport, and then they help with the shape and morphology.
Good.
For transport, this is the vesicular transport, which is like inside the cell.
Okay, good. So you have transport and then the shape. What is another function? It's related to shape.
It doesn't have to do with like structure, just having more shape.
Well, let me give you an example. It's a cylindrical shape. Cylinders are, loosely speaking, the strongest shape you can get because they have a maximized cross-sectional surface area, and that means that they can withstand a lot of weight. They can resist a specific type of force. What is that force that they resist? Well, let me ask you this. Loosely speaking, our legs are shaped like what? Cylinders, as is our arms. When I'm doing this, that's like 3 or 5 times my own body weight for like in terms of the PSI. What shape is it and what type of force is it resisting whenever I do that?
and then they're trying to go off.
You're going to be so angry when I get this to you. So right here, resists compression. Compressive forces, squeezing forces, pushing forces. All right, good job. Okay, so for transport, we have, keep going. And talk out loud to me as you're doing this. Walk me through it like as if I didn't know what I was talking about.
Well, for extracellular, there's cilia and flagella because they move as the cell itself moves.
Which one moves a cell itself?
Flagella.
Flagella.
And then cilia doesn't move the cell itself, but it moves things.
Move stuff around the
The
cell.
cell.
Is the flagella in a eukaryotic cell, like a sperm cell, is that a propeller type motion?
It's a dolphin type motion. And
Good.
Cilia's, not, it's a dolphin type motion too.
Good, they're both just bending and waving back and forth. Why do you think Dr. Dini cares about that?
Because that's its structure and function, is about structure and function.
Good, very, very good. What type of motor proteins help with vesicular transport or organelle transport, and what type of motor proteins help us with cilia and flagella?
Kinesis help with vesicular transport and creating and move organelles, and then dynesins help with cilia and flagella.
Good. Sometimes call them dyneins. I think it's called dyneins. I don't really know. Who cares? As long as it ends in IN,
Yeah.
and you know that means that it's a protein. Kinesin tells you kinetic energy movement. Dyneins means dynamics, but that doesn't matter. Okay, let's go into their structural arrangements that they can make as well. At the subunit level or at the monoscale, what are they made out of?
Tubulin.
Tubulin. Good. That's a conveniently named protein, by the way. Which, just venting with you here, enjoy that. When you're an undergrad, the names of the proteins make sense. Tubulin builds, the microtubules, right? When you get into other classes, they basically ran out of ideas for clever names, and you have these stupid CD195, you know what I mean? Like these stupid.
I think we're talking about in 20, talking about cancer, I think it's like 25 or something.
Yeah, there's a lot of really irritating names. Like I said, there's no systematic nomenclature in biology, so you can name things whatever you want to if you're the guy who discovers them, which is really irritating. Okay, so for the large scale, we have a 9x3 pattern arrangement, and the other one is a
9x2 plus 2.
good. The 9x2 pattern arrangement makes the
Cilia and flagella, and then just circle, and then 9 pairs of stuff, and then 2 in the middle.
yeah, and your textbook has a really good picture of this if you're curious about it. The 9x3 makes up something else. What does that 9x3 make up?
Centriole.
Good, the centrioles. If I have a bunch of centrioles together, what do I have?
Centrosome.
Good, centrosome. If anything ends with some in biology, what does that mean?
It means a bunch of something.
A bunch of something's coming together, good. The function, and I should have, whatever. The function of this, though, of the centrioles forming the centrosomes is the what? Right there.
Like microtubule organizing center.
Good, microtubule organizing center. Wonderful. Okay, if you don't mind, can you erase this for me, and then we'll get into intermediate filaments. Microtubules are the most time-consuming to do as far as information goes. They do a lot of things. It's okay. We'll just abandon the green marker.
It's like stuck on there.
Looks very ugly. Okay, so yeah, we'll just switch to the blue for the rest of our maps, and we'll do the content in red or orange. It doesn't really matter. Okay, so intermediate filaments. Let's talk about these both in terms of their structure and in terms of their function. So, structurally, what are they made out of?
They're made out of another protein too.
They are. I'll give you a hint. Your hair.
Oh, keratin.
Good, keratin. So intermediate filaments are made up of the protein keratin. And if you don't mind just taking a couple steps back, I know it's irritating, but keratin, you know, it's been so long since I've worked with kids, but there is this funny little like story that they told me one time that, hey, did you know that when you die, your hair and fingernails keep growing? Do they still say that? Are you familiarized with that myth? No. Okay. Well, people say that if you die, your hair and fingernails keep growing. That's not true. What it is these proteins are really, really stable and really, really strong. And so when you're dead and everything else is kind of rotted away, your hair and your fingernails do not decay. So you'll see these dead bodies with everything kind of shrinks after you die. It makes it look like your hair has grown significantly longer than it actually is. So, yeah, I don't know why kids would talk about that. Keratin is a super, super stable protein. I think all he wants you to know is just keratin. I don't think he goes into much detail on the filamental structure of it. But what is the functional role of intermediate filaments?
They help with structure.
They do, but how do they do that?
I just need structure.
Okay. Did you ever see the movie Pirates of the Caribbean? Okay. So there's this weird part where he's like, I made a raft out of my own hair. Like, where he says he's on like an island. I watched that movie a lot as a kid, that's why I'm saying this. But he says that he made a raft out of turtles and that he could use his hair as rope. Hair is actually pretty strong, so it's like rope. What type of force would you say rope resists? If I were to take your hair, I'm not going to, but does it resist compression?
Yeah, but it resists.
Okay, well, we'll use this as an example. Does this resist compression? No, right? I can squish it. If I push on it from the outside, it's not resisting that for anything. But does it resist?
Oh, it has elasticity.
Does it resist tension? Pulling forces, yes. So intermediate filaments are really good at resisting tension, and that's what we're going to put here. So something you may have noticed, and I'll talk about that later, that's pretty much all I'm going to say about intermediate filaments. Does he want you to know examples of this, of intermediate filaments in terms of the cell?
No, we just been talking about microtubules.
Yeah, microtubules kind of steal the show. But anytime you have the context of a cell, like for example the nuclear lamina, the cortex, you want to have some type of a filamental type structure. That's pretty much all I'm going to say on intermediate filaments. You'll learn more about those in the specific context of the body. Maybe you learn about something called desmosomes in anatomy and physiology. These are basically just really strong connections so that the heart doesn't tear itself apart whenever it contracts. Now let's talk about microfilaments. And both intermediate filaments, meaning that they have a rope-like structure, and microfilaments resist tension because they're filamentous, they're rope-like. But what do microfilaments do functionally?
I think they also have structure too.
They do. So what we're going to do with this, because this is kind of something that we're going to talk about a lot, we're just going to draw a big arrow down here and we're going to detail the functions of microfilaments. So yeah, like just write function. I'll do the math there. Okay, so what are some functions you can think of for microfilaments? If you don't mind just saying.
Again, structure, and all we talked about just the structure, but they have a structure.
They do, but they provide a secondary role with they do help structure. What are some structural contexts where microfilaments would be found in a cell? Okay, the cortex, sometimes called the inner cell matrix, would be an example of that. So go ahead and just write ICM next to function for the next map. We'll just branch right here and see the intercell matrix. else are some functions that microfilaments play a role in? Okay, that's fine. Did he show you guys the video of the bacterium being chased by the neutrophil?
Someone with the bacteria being chased by a
Yeah, yeah, neutrophils work. Yeah. So let me actually look at your notes and see, because I could talk a lot about this. Show me in your notes where he mentions, okay, so yeah, see, there you go, nuclear lamina right there. This is in your notes. Can you show me your notes on the day that he showed you all that video?
The white blood cell video.
Mm-hmm.
So I just have like the text of this one.
He's not mentioning much on this. Kind of irritating. But like I said, there is a gap now in terms of like the information that he's giving you and what he's going to test you over. So I'm just going to give you two examples of this. Microfilaments, their functions, there's really just two functions that they have. Amoeboid motion and then contractility. So we'll just do contractility and then amoeboid motion is the next two big points on our concept map. Good job. And if you don't mind, and I usually don't stress spelling of things, but oid, O-I-D. And the reason why that's important is anything that ends in oid, that's another one of those science terms that has a meaning to it. If I were to say that I got abducted by aliens, and those aliens were, they stood on 2 feet, they had a face, they had eyes, similar to ours, I would describe those aliens as humanoid aliens. If I were to say I got abducted by aliens and they looked like insects, I would say that those were insectoid aliens. So anything that ends in oid means that it's similar to. Amoeboid motion is the motion similar to an amoeba, a little tiny like bacteria. Okay, and so this is kind of hard to explain, but amoeboid motion, have you ever owned a hamster?
Mm-mm.
Yeah, I don't know why. It seems like the 90s was when small animals as pets was a big deal. Have you seen a hamster inside a ball?
Yeah.
Okay, how does that hamster move?
It just moves in the ball.
Hamster pushes on the ball, and then the ball
Moves.
rolls forward with him. That's basically what amoeboid motion is, but in this context, we're doing it in the cell. And what's pushing on it is not a tiny little hamster, but rather the individual microfilament subunits that are polymerizing in a specific direction, enabling that amoeboid motion to happen. So, amoeboid motion, let's just define what it is real quick. This is a way for the cell itself to move in one direction. Just say that this is how the whole cell moves. So in that video you saw of that white blood cell, that huge, massive white blood cell, tracking down that tiny little bacterium, he was able to do that because his amoeboid motion is actually faster than that bacteria's flagella could take in. Okay, so in amoeboid motion there are two things that are going to happen. Obviously we have polymerization to that end. So just go ahead and up here in the corner draw me a circle as a cell. Okay, so when we saw that video, let's say that the bacteria is over here, he releases molecules that this guy can detect in this direction. And what this cell is going to do is it's just going to automatically start polymerizing fibers, microfilaments, to push up against this cell here. And what that does is, if you can imagine a hamster in a ball, if we push up against something, it's going to kind of bulge out. And we can actually see that in the video that we see. There's this bulging out that happens. That bulging out is called something called a pseudopodia. And that is what we're going to mention for amoeboid motion. So what happens is the pseudopodia form, and being a little pretentious with spelling, but it's spelled, I don't know why it's spelled this. It's spelled with a P. P-S-U-E-D-O. Pseudo. E-D-O, yeah, and then podia. P-O-D-I-A, thank you for asking. Have you ever hurt your foot?
Mm-hmm.
Okay. May or may not have had to see someone called a podiatrist. So any tiny thing with podia means feet. And the reason why I'm mentioning this is eventually the pseudopodia form, and just how we move, our feet come first, and then the rest of our body follows. In cells, the pseudopodia sticks out, and then the rest of the cell follows. So you can actually know the direction that the cell is trying to move in based off of seeing those little pseudopodia form. Okay, and this is super important, by the way. And he'll talk about that in the context of the transduction pathway. I think he may already have, but we'll go into that a little bit later. Okay, so if the function is contractibility, what we're going to mention now is, by contractibility, I mean literally just that. So whenever you're moving your muscles, there's two proteins. One of those is involved in this, is microfilaments. The other one is called myosin, which I think you mentioned earlier as well. So let's just define what these are structurally really quick. Structure. So we'll just define that as a term by itself here. So they're all made up of a tiny little protein subunit called actin. So we're going to do two arrows obviously underneath the structure. The first one we're going to say actin. And actin is the tiny individual proteins. And it ends in IN, so you know that it's a protein, right? Okay, so there are two, we're going to use the word polymer structures for this. What does poly mean?
Poly is like
Many. And then mer means parts. So the polymer form, that means the form that consists of many parts. Okay, so go ahead and write polymer, M-P-O-L, oh no, sorry, no, not there. Right here, polymer. By the way, you're very good at concept mapping. You have a bit of an intuitive basis for it. Have you done these before?
Mm-hmm.
Okay. So there are two polymeric states in which actin can exist in, or microfilaments can exist in. There's F-actin and then G-actin. Good. Thank you for putting that hyphen there. What do those mean? Do you know? F and G, F-actin or G-actin? Okay. F means filament, so like a string, like your hair, and then G means globular, like a ball. Okay. So if I were to tell you that some type of a contractile force was needed to separate two cells and mitosis, which cytoskeletal protein do you think you would be using? A contractile protein would be? Yeah. So you may learn about a contractile ring later on. And in any type of a context, you'll have these things. Just a question, which do you think is better at resisting tension, microfilaments or intermediate filaments? Okay, why?
Because they can also have this contractibility. And so
Ooh.
then so if you contract, there's if you're like this, there's tension.
Yeah, I actually like that you're thinking that way. Let me just say this though. There is, let's say, you're not majoring in Spanish and math and chemistry and biology at once, right? You pick a specialization. It's physically impossible for one person to be an expert in every single thing on the planet. What we have is we have one person becoming an expert on just one thing. And that's what this would be an example of. Division of labor. If you can divide up your labor in terms of the job that you're going to do, you're better at what it is that you do, if that makes sense. So microfilaments, do they do just one job? No, they play a structural role in the cell matrix. They are contractile in the nevoid motion, and they do all this stuff here. So they are not as good at resisting tension as intermediate filaments. Intermediate filaments are designed to do one single thing, and that is resist tension, right? Microfilaments can resist tension just by their design, but structurally speaking, they're able to do a whole bunch of other stuff. Okay. Do you have everything for this? Are you able to save it, pictures? How exactly does that work?
Well, I could take a picture, but sometimes it doesn't like show up. Like,
Okay.
so it's just easier to take a photo.
Okay, sure. Okay, now let's. This won't take us as long. Okay, fill in the blanks for me, please. And I think I've said this, but when you're doing these at home, do this as well. The reason why this is helpful is you're given part of the information, and I'm willing to bet that when you take your exam, you're going to at least know some of what's going on it, but you're going to have to use your brain to fill in the blanks, and that's why this is helpful.
Are like hair. Keratin.
Good.
And there's this tension. So we do microfilaments and their function. And contractile.
Good.
And then, I know this is pseudo-simple.